Thank you all for attending this dissertation defense of Robert Del Carlo, PhD candidate in the Interdisciplinary Graduate Program in Cellular and Molecular Pharmacology and Physiology, or we call it CMPP. So my name is Norman LeBlanc. I'm a professor of the Department of Pharmacology at UNR School of Medicine. I served as Robert's co-advisor with Chris Feldman, associate professor in the Department of Biology and College of Science. We'll also speak a few words after me. So before I get going and introducing Robert, I'd like to provide a context for the audience on how this really exciting project came about. Chris and I first met around 2012 while playing scrimmage basketball in a Lombardi gym. And for a guy who was born in Canada with skates in his feet and not a basketball in his hands, that was quite an accomplishment. But so Chris was really the star. So, you know, since we were the only two lone older guys playing with college kids, we decided to have a little chat afterward and to ask Chris about his position, field of study, etc. And he told me that he's an evolutionary biologist. I didn't ask him about the evolution of what. He answered by saying that he studied the evolution of transmembrane proteins called voltage-gated sodium channels in garter snakes. Wow, I've been studying these channels all my life, you know, using biophysical approaches for over 30 years. So he told me he had done his postdoc at Utah State University, interacting with a good friend of mine, Peter Rubin, who is now in Vancouver, BC, an expert on biophysical properties of the ion channel Robert will entertain you with today. So this got the ball rolling, and basically Robert knocked on Chris's door. He was looking for a lab to do a research project in neuroscience, I believe. And so we set up a meeting with Robert, and immediately both of us were pretty flabbergasted by his intellect. During that meeting, it struck us that Robert's, you know, eloquence and intelligence, and he also had some ideas and thoughts about devising new therapeutics to treat schizophrenia by targeting another type of sodium permeable ion channel in the CNS, the NMDA receptor. And his long-term goal was to become a neurologist or a neuroscientist, or, you know, something related to neuroscience. So during the spring and summer months of 2013, Robert played a key role in collecting preliminary data, which allowed us to submit a grant proposal during the fall of the same year. The grant was funded for 4 years by the NSF and supported Robert's research starting in 2014. After graduating with 2 bachelor's degree in neuroscience and biochemistry, which is quite an accomplishment, Robert was admitted in the molecular bioscience program at UNR starting in the fall semester of 2014 and committed to the CMPP program late in 2015. So when Robert embarked on this project, Chris and I knew that he had to climb 2 large mountains with many foothills and valleys. So the first mountain, and that's the one I'm going to talk about, he climbed was to learn ion channel electrophysiology, both technically and conceptually, which is not trivial for most people. He devoured textbooks and papers in record time to truly understand and master what he was doing and why. He spent more time in my lab in these early days, and I had the great opportunity to monitor his progress. And I'd like to highlight some of his greatest qualities. Robert has a multiverse brain. So this is a popular word right now in astronomy, but he really does. He's interested in all kinds of topics. He's fun to talk to about anything, not just science. He speaks and writes 5 or 6 or more languages, including French, and not the least Chinese. He's an amazing independent learner and worker, and I can tell you that the wheels had to come off, the training wheels had to come off the bike very, very early. He's excited for science in general. He's like a little kid learning new stuff. One of his greatest qualities is creativity. I'd like to share my screen if I can. Which screen do you see? Let's see. Can you see this? No? Great.
Here we go. Yep.
Yep. Okay. So just to illustrate, this was sent to me by one of our secretaries. So this is the church in Virginia City. So Robert was born and raised in Virginia City. So he's a true Nevadan. And so he presented this model of St. Mary's Church to Father Joe. So here he was 16 years old. And he tells in this letter, and which I would like to talk to Robert about, but he talks about his friend and, that I guess made the model and all that, but shows you how creative, Robert is and was and still is. So this is, so creativity was definitely another thing that is a characteristic of Robert. He's also has tremendous problem-solving skills. So he's a true scientist, always thinking about new complicated ways to solve an experimental model. He had, just to give you an example, 4 data chapters in this dissertation and 7 appendices telling us what worked and what didn't, and for future students and projects, and millions of ways of transfecting cells to express voltage-gated sodium channels. He's also a fantastic teacher, and you will find this out today in his presentation. I think he will impress all of you. So he taught many of my undergraduate students. He's a great science communicator, and so here's an example. He made videos of all the techniques we use in my lab.
It's just rinse out with a little bit of water, mostly because even the tiniest little bit of salt buildup on your electrodes, as soon as you put your new salt solution in there, you're risking changing the ionic concentrations, and that is not only bad for your experiments, but even if it didn't harm your experiment too much, it could kill your cells.
So, and he's a great science communicator. He's a fantastic teacher, but he's a great science communicator. He won several prizes for oral talks and poster presentations at the local, national, and international levels. But in particular, he won the best presentation for PhD students twice at the annual 3-minute talk at UNR, and that led him to go elsewhere. So I asked him before he entered the competition for the second time, why he would bother the trouble and stress of going through that again. He answered, I need the money. So finally, I would like to say Robert is a kind and very empathic person, always willing to help others when he can with 100% commitment. And I'm truly proud of him, and I will miss him a lot. So I'd like Chris talk to you guys about the second mountain and other things.
Norm. I think I recall from that initial basketball meeting that you probably fouled out a few times because you play like a hockey goon. But we realized that we had each read and cited each other's papers. So that was really neat. I'll be really brief here because I don't want to cut into Robert's time, and I also don't want to get too sappy and have Robert get all the claim before his defense. But Robert has simply been a joy to be around, an absolute pleasure to advise and to mentor. And at times, Robert has even been a source of inspiration for me and other students in the lab. Robert is one of the most dedicated and hardworking and passionate students I've ever met. And it's his raw intellectual capacity, his depth of knowledge, and his creativity that have made him such a resource for me and Norm, and really make him such a bright young scholar. I think I've lost track of the times that the students and I in the lab have said, hey, we should get Robert's input on this idea. But one of the things I'm really most proud of for Robert is his ability to challenge himself and to learn evolutionary biology and to bridge the work in very disparate disciplines, neuroscience, molecular biology, and evolutionary biology, to help us make gains in the system that we're working on. So Robert, I want to thank you for entering our labs and our lives, for becoming part of our lab groups, for engaging in our respective graduate programs. And we have all benefited from the kindness that you show, the intellect, the dedication, and the generosity. Again, thank you all for joining us on this Zoom and for supporting Robert throughout this process. And we look forward to his talk. And we will try to hold a Zoom happy hour on Friday. I'll send an announcement around for that. So Robert, the floor is yours.
All right. Can you see me okay? All right. Well, thank you so much to my advisors for that introduction. So much for not getting my emotions riled up. For those of you who are used to my speeches, this one will be a bit longer than 3 minutes. Nevertheless, I'm very excited to present this research to you today. And I know I have friends all around the world tuning in, so go ahead and drop your location in the chat. Now, my research, as mentioned by my advisors, straddles several fields of science: biophysics, evolutionary biology, pharmacology, and toxicology. This means interdisciplinary scientists like myself must simultaneously tackle several ways of thinking. And it took me a half dozen years to try and grasp what's going on here, and I'm about to try and teach you everything in less than an hour. So I only have 2 words of advice: strap in. With no further ado, let's get started. Let's get started. Today we'll be talking about everything from proteins and populations and everything in between, and by the end, hopefully I'll leave you with some hope about predicting the impossible. So let's start off with a big question. Can we predict evolution? There's a lot of reasons that we might want to. This could help us sort out our ecology and our economy on several levels. But the basic question is it possible? Computational biologists working with virtual structures of proteins suggest that attempting to predict evolution is a fool's errand. On the screen, you're looking at our ability to accurately predict mutations. And as you can see, after the second lucky guess, our forecasting goes south. If this were a weather report, we'd have a pretty good idea of what's going on this afternoon, but not much far after that. So computational biologists say no, very pessimistic. So how exactly do we improve our predictions? I think the best course of action is to beef up our understanding of the past. And to do that, we need a good mental framework for retracing the steps evolution has taken. One of my favorite ways to think about evolution is like a species navigating a landscape through time. This framework is called a fitness landscape. And for now, let's just focus on one gene. I think we can think about this as a single strand of DNA from beginning to end. If we have 2 copies of that gene, we can square them up to make a matrix like we see in panel A. This is an example of a wholly fitness landscape, which is sort of like a piece of Swiss cheese. When you see land, that's a viable trait. Where you see a hole, that's a gene, sequence, mutation, trait, or combination of traits that would be an evolutionary deal breaker. So let's start making some mutations to our gene. We see a single point mutation that seems to fall on land, another one falls off, another one falls on land, one finds a hazard, another one seems to go a little off course, and then we have another one over here. Now, while the wholly fitness landscape is nice for working with populations, because any scientist can go out into any location, look at a sequence and see, okay, well, it exists. So obviously it's persisting in that environment, and what you don't see doesn't. But as a biophysicist thinking about proteins, I prefer not to think in binary terms. So let's add some information to this graph. If we add elevation, where greater elevation means greater performance in the wild, our fitness landscape looks a bit different. We can see that a species under selection is going to navigate its way up this fitness gradient. And from this, we can create some evolutionary trajectories from least to most fit, or worst, least performance to greatest performance. And we can get even greater information when we contextualize these trajectories by looking at the degree of competition in the wild. You can see that both of these trajectories work out pretty well, but as you increase competition, some of these trajectories may not be available. So return to the question, how do we improve our models of evolution? Next step, we're going to take clues from the animals living today. And let's start with these beautiful animals and the chemical they all have in common to protect themselves. It's called tetrodotoxin, or TTX, and it's a fantastic broad spectrum chemical defense because it shuts down the electricity in our bodies that we depend on to react to the environment. So just about anything attacking these animals will be facing a steep physiological challenge. Let's try and get an idea of how potent this toxin is. So if you had a sugar cube in your hand, that's 1 gram. If instead you had a 1 gram TTX cube,
you would be holding a lot of lives in your hand. Administered orally, 1 gram of TTX could kill about 43 NBA players. We're talking about 200+ pound adult beefcakes, and 1 gram could knock 43 flat. Administered intravenously, however, 1 gram of TTX could take out 25 NBA leagues, over 12,000 people. So you can see how this extreme physiological challenge would be something, would be quite something for anything to face, a very deadly chemical defense. And while these numbers might seem kind of ridiculous to you, they're quite paralyzing, because over the last 6 years, I've used a lot of this stuff. But don't worry, what you're looking at on the screen is only enough to take out 100 or 200 of you. So having a defense like this on board should really deter any predators. So given what we know about tetrodotoxin, its potency, and its natural sources, I'm going to show you something that doesn't make too much sense. Here, we're observing the common garter snake preying on a newt, which we know is defended by tetrodotoxin. You might think this is going to end poorly for that snake, but in reality, that little fella is going to be completely fine. About 60 years ago, my doctoral advisor's doctoral advisor, Dr. Butch Brody, there on the right, started investigating this very perplexing sight. What struck him most is that he couldn't help but wonder how something as small as a newt could possess enough toxin to take out 2 basketball teams. Well, this is how it works out. The newts didn't start off extremely toxic. Instead, in ancient times, they had a little bit of toxin, but any species that kept preying on them will experience that selection pressure, killing off any individuals that can't handle the toxin. But then, by some stroke of evolutionary luck, the snake started evolving resistance to the toxin. This redirected the selection pressure back onto the newt, which then in turn forced them to amp up their toxicity. We call this escalatory reciprocal adaptation a co-evolutionary arms race. After potentially millions of years of this interaction, today's newts are extremely toxic, while some of today's snakes are so resistant that no newt could produce enough toxin to paralyze them. But how exactly did these snakes evolve resistance to the toxin? For snakes, the answer was simple. If someone has the key to your house, change the lock. And this is that lock. This is the protein that is responsible for the electricity in our bodies that we depend on to respond to the environment. This is called a voltage-gated sodium channel, and it sits in the membrane of excitable cells. And when stimulated, it dilates a pore that admits salt into the cell. This is just normal table salt, sodium, that rushes into the cell when the protein opens and carries an electrical current that the cell can use as information to do work, like contract cells or propagate messages. So let's get to know this protein a little better. These are our sodium channel historians. Scientists have been studying sodium channels formally and informally for centuries, and it's proven to be quite lucrative. 80% of the people on this slide were awarded a Nobel Prize for their contributions to understanding bioelectricity. Poor Luigi Galvani there, he died some 35 years before Alfred Nobel was even born, so he missed out. However, we've only been studying sodium channels for a very small fraction of their very ancient history. Here we're looking at a phylogenetic tree showing sodium channels evolving some 800 million years ago. They're found in a huge branch of the tree of life. Looking at this tree, the most fearsome predators fall under the category of tetrapods down there at the bottom right. That includes us and our snakes as well. So it's no over-exaggeration to say that sodium channels opened up entirely new possibilities for life. Now that we know where we find these genes on the tree of life, let's see where they find themselves in our genomes and our bodies. There are 10 individual copies of the sodium channel genes dispersed throughout our genomes, and they've all been evolving independently within us and have become specialized to be expressed in certain tissues throughout the body. Today, however, we're only going to be focusing on the skeletal muscle version, NEV 1.4. Now let's get a better idea of the skeletal muscle voltage-gated sodium channel in its home. Here we have a neuron in yellow and a muscle cell in gray. When the nerve releases acetylcholine onto the neuromuscular junction, it activates acetylcholine receptors that in turn activate voltage-gated sodium channels. The sodium channels then take that signal and spread it like wildfire over the membrane in the form of an action potential, like that. As the action potential passes over the membrane, it excites another type of voltage sensor in the plasma membrane called the dihydropyridine receptor, or DHPR for short. This is physically coupled to intracellular channels called ryanodine receptors, shown here in orange. When the voltage sensor activates because of the action potential, it pops open those ryanodine receptors, which allows calcium to enter the cytosol and then have access to the contractile machinery of the cell. When exposed to calcium, these structures shorten, and this is the basis for locomotion. So now that we have an idea of where we find these sodium channels in life in our bodies and how they translate nervous input into locomotion, let's look at how these snakes have gone about tinkering with them. We'll focus on these blue parts here, which form the ionic pore. This is where these snakes have evolved mutations. You can see these ribbons represent more complicated structures, but we're going to focus on just one at a time. There are two main regions of the protein that we're going to be focusing on where these mutations have evolved. These are the pore-forming elements of what we'll call domains 3 and 4. In one species of garter snake called Thanophysis atratus, there are animals that have sodium channel structures that look like yours and mine and horses and flies and cheetahs. When nature avoids messing with the original recipe, we say that this is an evolutionarily conserved gene across phyla. But there are some populations of Thanophysis atratus that take this alanine at position 1281 within the protein and mutate it to a proline. This is our first mutation. This is our first deviation away from the recipe that works well for the majority of animals on Earth. We'll call this mutation P for short. There are another population of snakes that take 3 residues of the sodium channel and mutate those. We'll call this one EPN for short. In another species, Thanophysis totalis, the common or red-sided garter snake has some populations that only have a single point mutation. We'll call that V for short, while others have 2-point mutations, which we'll call VA. And then there's the Mac Daddy. Some populations of Thanophysis totalis have evolved a whopping 4-point mutations in their sodium channel pores, which we'll call LVNV. I like to remember that as Las Vegas, Nevada. So now that we know how these snakes have changed their locks, let's get some context into looking at where we find these mutations let's get some context by looking at mutations in human sodium channels. Essentially, this protein is just a ball of yarn. And if we give it a tug, we can pull it apart into one long string with a beginning and an end. Red X's here document where we find mutations in human sodium channels. To be clear, everything in red is maladaptive. In yellow, however, we see that's where our snake mutations crop up. So our question is whether or not these mutations have consequences for messing with that original recipe. This idea is not entirely new. We've seen that single point mutations can wreak havoc. We've seen this in serious diseases such as sickle cell disease. A single change creates a hiccup in a protein that results in unhealthy blood cells. But there is a little bit of an advantage here because someone who has one copy of the good channel and one copy of the bad channel it actually has some malarial resistance. So there is this positive pressure maintaining an otherwise deleterious allele in the population. So there's competing selective demands here. And we can think about this as like kind of 2 separate hills. And we're asking this is an example of nature asking too much of one protein. So I want you to keep this in mind while I build an argument for fitness landscapes in our snakes. So to do that, let's get started on the research that I did. Do trade-offs exist in the snake mute system? First off, we know that TTX blocks the ancestral pore structure. I should point out there that HS in the bottom line refers to Homo sapiens. And all those dots mean that we have identical structures to the snakes that cannot eat newts. Ergo, we as humans should not eat newts. However, in snakes that we've observed eating and surviving eating newts, we've observed these pore domain mutations. So our hypothesis is that these mutant channels are no longer blocked by such a toxin. So let's start by analyzing these sodium channels. To do this, we're going to employ this technique called whole cell electrophysiology. Essentially what we do is we take some cells expressing sodium channels and we put them at the bottom of a dish, cover them in a salt solution. We add an electrode to the bath. That's going to be our reference electrode. And then we're going to take this glass pipette and dip it into the bath and bring it up to the surface of the cell. We'll apply some suction to the pipette and to bring a portion of the membrane into the tip. Once you break a small hole into the cell, the electrode inside the pipette faces the inside of the membrane, while the reference electrode faces the outside. This gives us electrical control of the membrane and therefore the proteins in it. So let's take a look at what happens when we shock these sodium channels just a little bit. In this video, you can see that the sodium channel moves through several conformational changes once it is stimulated. Under normal circumstances, when the cell is at rest, the inside of the cell is very negative. And since opposites attract, positively charged residues in the sodium channel are sucked inward and downward by this electrical grip. When the cell is stimulated and the inside of the cell becomes less negative, this loosens the electrical grip on the cell, on the channel, and these positively charged residues can move upward, releasing them upward. As this happens, this conformational change on the pore of the protein allows for like a little bit of tugging that dilates the pore, just enough to admit sodium into the cell, seen here in green. We call that activation. Then later, that red portion of the protein down there at the bottom, that comes in to plug the channel from the inside, and we call this inactivation. Let's take a closer look at these processes. Let's start off with a cell at rest with a negative membrane potential of about -100. This is about 15 times weaker than a AAA battery. As the voltage stimulus becomes more positive, we see that the sodium currents generally get bigger and bigger. After a while, they start to get smaller, but don't get fooled by that because sodium itself is a positively charged ion. So as the inside of the cell becomes more positive, it's going to slowly repel it outward. But this doesn't mean that the sodium channels are closed. It just means we're registering a smaller sodium current. The channels are still open. When you account for this effect on the ion itself, we expect to see an activation relationship like this, where more channels are opening as you increase voltage. Now let's look at how inactivation is affected by voltage before they fire. The electrical field of the membrane can also determine the proportion of the channel population that is available to fire at any given moment. Before we were holding our cells at -100 millivolts and then we pulsed up. Here we're going to change what we hold our hold our voltage at before we fire. If we start off at -100, we can see that 100% of our channel population is ready to fire. However, as we progressively depolarize the membrane, that is, make it less negative, we see progressively smaller currents, which means fewer and fewer channels are available to fire. This is an important feature of sodium channels because they absolutely depend on a negative environment to function. Ultimately, we expect to see a relationship like this, where before the membrane fires, that voltage there will predict how many channels are ready to open. So here's what the currents from the ancestral channel look like when we activate them. Nice, fast, robust. However, we can see that sodium channels carrying mutations generate much weaker currents. Moreover, these mutations appear to recreate the resistance seen in snakes. A small concentration of tetrodotoxin can completely abolish the electrical current in the ancestral channels, while even double that concentration hardly has any appreciable effect on the resistant channel currents. If we look at the current voltage relationships, we again see that even at their maxima, resistant channels develop weaker electricity. However, this appears to be the extent of it because we don't see any changes to activation or inactivation across resistant channels. So to further investigate the mechanism of this reduced current, we're going to utilize a statistical technique. This technique is called non-stationary noise analysis. And just as that mouthful sounds, it means we're going to try to pull information out of noise. To do this, we're going to pulse the membrane and its sodium channels about 1,000 times to elicit 1,000 current spikes. In the middle graph, I'm recording these currents as single 1 pixel thick lines that are color coded to represent the strength of the current. As the procedure continues, our idea of the mean current gets cleaner and cleaner. That's the red line that's superimposed over those individual traces. Once we have a mean of all 1,000 traces, we can then subtract it from each of the 1,000 currents that generated it, and I can record those difference currents there in the bottom graph. These difference currents point out where each individual trace seems to deviate from the mean, which is directly proportional to how much each sodium channel is contributing to the overall spike. This noise is a rich source of information for us because it is related to the underlying molecular mechanisms. Essentially, if we take this white line here, which is the mean current, and plot it against the corresponding point on the dotted line, which is the variance or the strength of noise at that time, then we get parabolas like these that tell us about the strength of the underlying sodium channels. And here's how. Basically, it falls down to this principle. If everyone is doing the same thing, then the variance is 0. If everyone is quiet, the variance is 0. If everyone is shouting at the top of their lungs, the variance is 0. But if half the room is randomly quiet and the other half is randomly shouting, then we have a lot of noise to work with. And this noise relates the overall chorus of sodium channels to the solos of individual channels by a relatively simple parabolic equation. Allow me to draw your attention to the most important parts. The slope of the parabola is directly related to the unitary current. This is how much each individual sodium channel conducts when it opens. I'm famous for referring to this as how much sodium a sodium channel could channel if a sodium channel could channel sodium. Because the toxin-resistant channels have weaker macroscopic currents, that's the overall spike, without any other deficits, this is what we're interested in. That small I is what we're interested in, the steepness of that parabola. And we can also get other information out of this technique as well. So let's try and understand what's going on with these parabolas. These mean data here seem to replicate what we see at the macroscopic level. Toxin-resistant channels are simply weaker. They have smaller unitary conductances. However, this does not come at the cost of any statistical differences in the number of channels in the membrane or the maximum open probability, which is just another nail in the coffin pinpointing the sodium channel conductance as the smoking gun in this system. This is the source, the proximate cause of the trade-off. These mutations reduce the amount of toxin, or reduce the amount of sodium that can move through these things. So let's see if this trade-off scales up to the muscle level. To do this, we're going to do something called myography. We're going to take a skeletal muscle there, suspend it from a lever and on a platform. And then we're going to use those two platinum electrodes on either side to apply a stimulus, which is going to cause a contraction, and then it will relax on its own. This time we're going to record that information. We apply a stimulus, we have a contraction, which we see as a rise in force or tension on our graph, and as it relaxes, it decays back down. Now, let's look through some of my results in both of these species. First, we should establish that muscles from snakes that can eat newts are themselves resistant to tetrodotoxin. Check, they are. Then, let's ask if there's any difference in the amount of force these muscles can produce. Voila. Now, this is just one way to look at the force. Let's look now at tetanus. Tetanus is what happens when we apply a sustained high-frequency pulse and the contractions of the individual fibers begin to fuse and create a sustained tension, in which we see this trend persists. Now, while our snakes are not constrictors, this protocol should be an indication of their endurance and which would be important for capturing prey and fleeing or evading higher predators. So this trade-off is pretty stark. We've seen that toxin-resistant sodium channels generate less electrical current. We've also seen that toxin-resistant muscles generate less force. Let's check to see if there are any deficits at higher levels of organization. To do this, we're going to start off by measuring whole animal resistance. What we're going to do is we're going to motivate these snakes down a racetrack here and clock its baseline speed, how quickly it transects that racetrack. Next, we're going to start injecting them with TTX and rerunning them down the racetrack. Here, we're looking at a poor snake that just is not handling its toxin very well. We're going to take this and look for the amount of toxin it needs to reduce their sprint speed by half. And we're going to call this, forgive me, MAMU, or mass adjusted mouse unit. This is just a way for us to compare the doses for the snakes to the comparable dose that would kill a comparably sized mouse, okay? So if we take these scores and we plot those on the landscape, we see these distributions. Now, to my eye, if these mutations came at no cost, then we would expect to see these maps be entirely dark red or entirely dark blue, because these mutations would ensure that any snake anywhere could eat any newt it came across. However, we do see this heterogeneity, and we do see that these mutations appear to be clustered in specific spots. So that to me suggests that both the ancestral channels and the resistant counterparts have selection pressures acting on them to maintain them in the population. So to recap a little bit, weaker currents are associated with weaker muscles, which are associated with reduced geographic success for these resistant alleles. Why might this be? Well, in short, it might have to do with the compromise of these biophysical channels. It might be because our mutant snakes are compromised to the point that they can only persist in areas of the geography where conditions are relaxed enough to support a weaker phenotype, whether that is being susceptible to tetrodotoxin, which is not favorable in some areas, or having weaker muscles, which is not favorable in some areas. This is something that is true for all of life. We only exist on the good graces of a very chaotic and harsh world. And I should point out there that that snake being eaten is not itself eating a newt, but how cool would that have been? Too good to be true. So here's another big question in my research. We've already established that these mutations face trade-offs, but can these imperfections help us put together a model of how evolution proceeded step by step from ancestor to descendant? So let's start off by focusing on one species, Thanopus atratus. In this species, we only see 3 sodium channel alleles. We see populations carrying the ancestral channel. We see populations carrying a single point mutant, A1281P, we'll call that P. And then we see populations carrying the triple point mutant, EPN. Even more interestingly, we seem to observe that these mutations exist in a progressive pattern over the landscape. Just looking at this map, I'm inclined to think that the ancient toxin-sensitive channel might have had like this stepwise position where A1281P is a stepping stone on the way to the triple point mutant EPN. This is an attractive idea because we don't see any other intermediate mutations on the landscape. We don't see E by itself, we don't see N by itself, and we don't see any combinations thereof. So let's see if we can make a little bit more sense of this. Let's just assume that the ancestral protein, the DAD, the toxin-sensitive channel, is older, right? We'll put that up at the top. And then the triple point mutant is the descendant. If the need for toxin resistance is driving the channel structures to evolve, then there are only so many steps between the separating these two. There's only so many evolutionary paths through Darwinian space that can take us from the ancestral to the triple point mutant. So here we look at these paths, and there's 6 possible paths from DAD, the ancestral channel, to the descendant EPN channel. 2 paths running through each single mutant, okay? Now let's dive into the muscle physiology of those of these that are extant in snakes and see if we can eliminate some of these paths. Here we see the skeletal muscle twitches of each genetic population. Each population is associated with a progressive reduction in force. We see this is repeated in how quickly these muscles generate force. We also see this in the tetanic contractions and their rate of force development. And we can see that a single point mutation offers a moderate amount of TTX resistance, while the triple point mutant offers extreme resistance. You can see these trends where increasing mutation number is associated with increasing resistance, while increasing mutation number is also associated with decreasing muscular force. So this is the crux of the trade-off. Each step away from the original recipe puts you in a little bit of jeopardy. Let's look here for more context. So a little bit of associative evidence here. We only find that D1568N in snakes that are extremely resistant and extremely biophysically compromised. So on merit alone, I'm inclined to think that D1568N is a very costly mutation that snakes can acquire under extreme circumstances, but they'll have to dig themselves out of that biophysical hole pretty quickly afterward. So let's walk through our evolutionary trajectories here. There are 6 possible trajectories from ancestor to descendant. We know that the only intermediate mutation that persists in nature is A1281P. So it seems that the 2 most likely paths will proceed through that first P mutant. Moreover, we do not see any populations that carry only D1277E, which suggests that a toxic diet alone is insufficient to drive it into existence. But we do know that D1568N is the only mutation that is common to both extremely resistant snakes, and we also know that it's common to muscles that are extremely compromised. So putting it all together, I suggest that in Thanopus atratus and ancestral snakes first evolved a single point mutation, A1281P, which provided sufficient resistance for a lot of the habitat range. However, when facing extremely toxic newts in the south, the only way for the P population to meet this challenge was to take the extreme biophysical hit, incorporate the costly D1568N, which does improve TTX resistance at the cost of performance. But this reduced the performance so much, and that reduced performance served as an intrinsic pressure that drove the existence of D1277E to fine-tune the structure of the pore and perhaps restore some of that biophysical performance. So you can see how I'm starting to think about this evolutionary trajectory as a sort of fitness landscape. This last big question of my research is how these mutations interact with the landscape. These channels, as we just saw, they seem to bumble through sequence space, bumping up against their biophysical limits. So if these channels take the path of least biophysical resistance to greatest toxin resistance, then surely some of the resistance, some of the forces keeping them on track come from the very environment in which they are evolving. So let's focus now on Thanopus ortalis for this part. Here's what the ancestral muscles can produce. Single and double point mutants introduce a little bit of variability into the contraction while also moderately increasing TTX resistance. And then there's the LVNV snakes that are badly affected despite the adaptive value of extreme TTX resistance. If we look at the geographic distribution of these mutations, we see two pretty obvious trends. First, the mutations seem to have this relationship with, the mutant snakes seem to have this relationship with latitude that ancestral snakes do not, where greater resistance is found in the south. Another gradient that follows this trend is temperature, where warmer climates are also more south. This is, to further investigate this coincidence, let's go back to the lab. Here, we're going to use the myography bath again to assess these muscles. Essentially, what we're going to do is we're going to apply a stimulus, and each time we're going to increase the amount of current running through that stimulus and see how much it takes to cause a contraction. Here, we're going to be looking for how much stimulus current it takes to get the muscle to about 10% of its maximum, and we'll call that our threshold. And then we'll repeat this at 6 different temperatures. Now, intuitively, if you've ever tried to tie your shoes in the dead of winter or tried to untie them in the middle of a heat wave, then you're familiar with the effect of temperature on muscular activity. Looking at this threshold, that is how much energy it takes to activate these muscles, we see that single and double point mutants are not really affected relative to the ancestral TTX sensitive or TTXS muscles. However, the quadruple point mutant consistently requires greater energies to activate. So looking at that more closely, there on the right, we see the activation curves for these muscles. And at colder temperatures, we observe that the resistant snakes are severely hypoexcitable. This suggests that extremely resistant muscles don't perform very well in the cold, and that these animals, I mean, they're ectotherms, and they rely on the environment for temperature regulation. So this could be problematic. So let's look at where we caught them to see if we can get some information from that. Here we have the west coast of the United States with the minimum temperature in these areas color-coded over the geography. The blue dots indicate where we've sampled the snakes. And if we pull climate data from each point and we look at if there's any differences between resistant and ancestral snakes, we might be able to get a clearer idea of the habitat profiles of each genotype. So if we look at elevation, we see that resistant snakes are often found at lower elevations, which tend to be warmer. If we look at precipitation, we see that they're in areas where there's less precipitation, which also means it is likely to be a warmer area. We see greater sunlight in the areas where we find these snakes. And if we look at the maximum temperatures in those areas, the mean temperatures in those areas, and the minimum temperatures in those areas, they're all consistently higher for the snakes with these 4-point mutations, which suggests that they only persist in areas that are warmer, which is consistent with what we found in the muscles. So doing that, having established these habitat profiles for the animals, what we can do is a little bit of forecasting. We can take those profiles and look at all the areas on the landscape where these would be suitable habitat for each group, okay? So doing that, for LVNV, we see that the brightest areas, that's the area of the landscape where LVNV snakes are particularly well suited to live. Now, if we look at how much of the environment the ancestral snakes can use, we see this, and this to me is night and day. These ancestral snakes have a much larger playground than their toxin-resistant counterparts. They have their profiles fit a large proportion of the geography. So if we subtract one map from the other, we can really get a sense of that. And you can see the areas bounded in red. That's the limits of where LVNV snakes can really persist, implying that the ancestral sodium channel with its areas, it just goes so much farther beyond that red, and suggests that maybe it performs better over a wider array of conditions, which might have implications for fitness and the longevity of the species. So now that we have seen that nature is asking too much of these proteins, we know that doing so will force a protein to come up with imperfect solutions. But these proteins are so important throughout the tree of life that each step through evolutionary space is consequential, and their impact on performance may be strongly related to fitness. So let's see if we can gain some evolutionary insights from these broken channels. The relationship between such an important protein and the survival of the animal helps us make sense of the metaphorical fitness landscape we're facing. The main trends that we see from this research is that the ancestral sodium channel represents a peak in the performance of two snake species, two separate snake species. When these snakes are put between a rock and a hard place, they will acquire mutations that make for imperfect solutions to an impossible problem. However, it is interesting that the apparent order in which they do so seems to butt up against these biophysical limits. So bearing in mind that the structure of the channel, when you make changes to it, even for a good reason, it's going to have consequences. Let's see if we can recreate a fitness landscape for Thanopus species. Here we see a putative fitness landscape for the Thanopus genus. I've put the ancestral channel up at the top, marked with a plus sign. For Thanopus atratus, we see that the most likely first step is A1281P, because that's the only intermediate mutation that exists on its own in this species over its range. For Thanopus atratus that needed to acquire greater resistance, their only viable option appears to pass through that performance valley where they can pick up D1568N, which is likely to be costly to performance. Then they climb out of that hole by fine-tuning their structure with D1277E. For Thanopus sertalis, we know that every population that evolves resistance in this species begins with the same first point mutation. That's I1561V. We know that from ancestral gene reconstructions by Mike Haig. From here, though, there appear to be two divergent paths. Either an additional single point mutation can be adopted, that's G1568N, or if extreme resistance is needed, Thanopus sertalis likely drops into that performance valley to pick up D1568N, which is the same from Thanopus atratus, which we know is very costly but very effective for TTX resistance. It will then have to climb out of that performance valley by subsequent mutations, and we don't know the order of that yet. I would like to point out that the metaphorical water level for Thanopus sertalis going the LVNV route may have been lower, and this might have to do with the temperature of the environments in which they evolved. It might have been the very environment that supported this evolutionary trajectory that biased this population of snakes down the LVNV route as opposed to the VA route. So let's return to the big question. This notion of predicting evolution. I said from the start that the best way to predict something is to understand what has already happened and how. We've just walked through a few impossibly complex eventualities that natural selection has sorted out. And here are the lessons that I think we can walk away with, at least some of them. First, asking too much of one protein will force it to make imperfect solutions. Highly conserved proteins, therefore, make extremely great models for reconstructing evolutionary trajectories because their recipes don't change. And when we do see changes, they have obvious, readily interpretable implications. And lastly, no mutation exists in a vacuum. So to really get the full breadth and nuance of an evolutionary trajectory, you have to look at the environment in which these evolved. Now, while we as scientists cannot outwork evolution, we can outsmart it. In just this one system, studying just one protein at several levels of organization, we have built arguments of how stepwise evolutionary change may have proceeded in two species, and how the environment itself may have expanded and contracted the evolutionary possibilities available to certain populations. Now, I don't mean to be cocky, but I'm thoroughly confident that within my lifetime, we will be able to accurately predict many mutations into the future and use that information meaningfully to preserve and expand life. Now, we're only at the precipice of being able to synthesize all of this known biophysical information, all of these rules that will one day make predicting evolution as reliable as the weather report. What we have learned from the snake's sodium channels is manifold. But ultimately, we know there's no such thing as a free lunch. These mutations came at a cost, and these snakes have had to bumble their way through evolutionary space to arrive at the mutations that we see persisting today. But where do we go from here? These snakes and newts have been so bitterly embattled among themselves. All the while, the world is changing around them, and they may have potentially relied on that changing environment as a crutch to support failing sodium channels. As the world warms around them, some populations of snake species may find that their suitable habitat expands, becomes more abundant. Alternatively, it's possible that these snakes, which exist in California, which catches fire every August, may face some problems because they have reduced locomotor capacity and may not be able to escape some environmental threats. So maybe they put their eggs in a basket that catches fire every August. So I'd like to leave you with the idea that this is still an evolving system and that the future may portend grave tragedies or great opportunities for these snakes. Now, speaking of the world, I won't get a traditional commencement this year because of an abundance of caution for coronavirus as it is. So I want to take a couple of moments to celebrate some of the parts of this journey that I won't get to celebrate otherwise this year. First and foremost, because of graduate school, I'm almost as well traveled as COVID. For conferences and through the friendships I've made in these last few years, I've gone all over the world, and I am so grateful because this image, more than anything, is a great thank you to my advisors, Norm and Chris, who have supported my work on an international platform, and I am grateful for that. Also, I'd like to take a quick shout out for the UNR 3-minute thesis program for getting me out of debt twice. And also I wanted to extend thanks to Nancy Maldonado and Susan May Smith at PBS Reno, formerly KMPB, who gave me a platform to bring science careers and my graduate education into Washoe County classrooms. And a thank you to all the teachers who welcomed me into their classrooms. I've been told a couple of them might be watching today. And as their student to later bring my information into their classrooms as a guest lecturer, I went into the very classrooms where I once was a student at DeMonte Ranch High School. That was very meaningful to me. And then there's this photogenic bunch. Believe it or not, most of those people are already or will soon be doctors. So thank you for everything, y'all. And then a quick shout out to Jana Magana and Angelo Lippi, who have been my most cherished students and friends over the last few years. Jana was instrumental in chapters 3 and 5, while Angelo's work was critical for chapters 3 and 4. These two have been by my side even when the lab was literally falling apart. And I want to give a shout out now to someone who pushes me, who supports me, who never fails to see the positive qualities in me, even when I'm not trying. She's the fittest person I know. She floats like a Lepidoptera. She stings like a Hymenoptera. She's the newly minted Dr. Jessica S. Remke. In addition to all these wonderful times we've spent together, when she first met me, I seriously could not even touch a snake. I had a visceral comical reaction to them. Norm LeBlanc is famous for calling me Indiana Jones for my hate of snakes. For example, this is the first time I ever held a snake. And that is not the face of someone who likes their job. But through Jessica's own version of persistent exposure therapy, this next picture was taken just this summer, me out in the wild catching my own animals. So thank you, Doc. I owe you a lot. Then there's my family. we're a modest people. We've never had a lot, but these people have made sure that I will never be poor. I am so thankful for all of them. My first job, my family, an endless source of positivity. Thank you so much. This year sucks. You can't imagine how hard it was to watch the world literally fall around you and still try to put together a dissertation while all of your heroes seem to be dropping off. It was hard, and I commend all the students that did it. This thing sucks too, but I look at this image with great pride because this year, the University of Nevada-Reno Microbiology Department sprung into action to get us on the right foot with this pandemic. And I really was so grateful and so proud to see my friends pop up in my newsfeed. You're all rock stars. Another positive thing, lighter news, because I need it right now. This was a great year for scientists and thinkers with 2 X chromosomes. While the work I was discussing here today in the last hour has principally been focused on reconstructing the paths evolution has already taken, Doctors Emmanuelle Charpentier and Jennifer Doudna have been recognized this year for the 2020 Nobel Prize in Chemistry for giving humans the ability to outsmart evolution in ways Charles Darwin could have never dreamed. And not to brag, but I took this picture of a future Nobel laureate. That's the closest I've ever been to royalty, and I have been to Buckingham Palace. And lastly, I want to thank so many people here. You have been my allies, you've supported me, and thank you for getting me through this journey. And that's all, folks. Let's see if we can take some questions.
Wonderful, Robert.
Thank you so much.
Incredible.
Lots of kudos here coming in. Any specific questions? We're happy to take those. Robert is happy to take those.
I have a question, Bobby.
Yeah, who's speaking?
Amanda.
Oh, hi,
Yonan.
Amanda.
Hi, you did a fantastic job. That was wonderful. Because I know that you're so interested in humans, I'm going to ask this maybe sacrilegious question. How do you think you could use this research to help human phenotypes?
Oh, I love that you asked this because you know how much it riles me up. Well, in short, it goes right back to things like the coronavirus. Here we're looking at a novel challenge for us as society, as a human race. This molecule, this organism, this thing, this virus, it's going to evolve, and we've seen this with the flu. We see this with a lot of our health challenges, our pathophysiology that deals with bacteria. Evolution is this thing that we're constantly trying to follow up with. So if we were to, look at this this organism or look at any biological challenge like the coronavirus, like antibiotic resistant bacteria, if we can identify the trade-offs that are too costly for those organisms to sustain, if we can identify the places where the physics of those organisms are going to rein back the ability of those organisms to survive, then we can use that to our advantage therapeutically. I'm not a microbiologist by any means, but it could be down to the very folding of the proteins that constitute this organism, helping us outsmart us. So that's one application. There's a bunch of other ways that you could use toxin resistance to study human research. It's a very powerful thing if you can use put something that sustains electricity in the presence of a toxin. You could use that as a great research tool. For instance, putting it in some neuronal cells in the brain, applying toxin, the tetrodotoxin to the brain, and then only observing the cells that can still fire because of the sodium channels that you've given it. That would help you narrow down certain areas. But I think those are two generally broad answers. If that suffices.
Sorry.
Yeah, may I ask a question?
Yes, of course.
Hi, Robert. That was wonderful. That was really, really good. I'm so glad I tuned in for this. So we saw this week. So the exciting news this week was that we may have an AI system that would allow us to predict protein structures. We'll
Yeah.
see. But how do you think, so you're talking about using, you know, your research as a way of possibly predicting future mutations. Can you see any way that you could couple those two things together? So you say, okay, I'm going to say maybe this mutation, and we may be in a position now where we can say, oh, and this will be the structural effect. So,
well, I am so thrilled that you asked that question, because I followed that research as well. And even before this week's research with artificial intelligence, there was something that came out in February the year that was equally exciting. While my research does tell us to focus on the individual point mutations and their local structural effects, coupling those rules to what we would find doing these broad spectrum attempts at the at AI could be very powerful. And here's an example of how. So what came out earlier this year in February is that basically this group at MIT, they took a library of antibiotics that they know have certain effects on antimicrobial effects, bactericidal or something like that. And they gave it to a computer. And so they asked this computer to, write down its own rules about how, what chemical motifs are associated with what effects on bacterial viability. And after a few days of letting it write down its own rules and come up with its own algorithm, they then fed this machine some 100 million chemical structures and asked it to think about what could possibly come up, which chemicals might fit the brief of these new rules. And they came up with a new chemical called halicin. It's been around for a while, and it has a very specific molecular function. But this halicin was predicted by this algorithm to be a very good antibiotic. And not only did it turn out in actual bench experiments to be a very good antibiotic, it was extremely effective against many antibiotic-resistant strains of bacteria. So this, to me, I think of proteins as nothing but extremely large chemical structures. So what I would like to do is take a sodium channel, for instance, just the 80 or so poor residues, and make a bunch of mutations. Start at each mutation, cycle through all of the other 19 amino acids that it could possibly be, throw TTX in there, calculate the affected binding affinity of those two molecules, and let an algorithm cycle its way through something like that to point out the evolutionary trajectories that may lead to a sequence, a channel sequence that has the best TTX resistance, but that we don't see in nature, seeing a lost opportunity that nature didn't take, which we would then probably follow up with to see if there's an extreme trade-off to such a mutation, that it's just too costly for mutants to sustain. So I think the use of artificial intelligence, like this algorithm, like the one you're talking about with the predicting the protein folding, I think that could be extremely helpful, because one of the main things that we're missing is that structural context. So combining what came out in February and what came out in this week, I think we're going to make great strides in being able to predict what molecules will do in the face of certain challenges.
yeah, thank you.
Thank you very much.
Can I jump in there, Bobby?
Sure, come on in.
Hey, so maybe just to follow up with Dr. Ellison's question. So oh, by the way, you did an awesome job. Sorry, I should start with that. I'm sorry, I just was really interested. So, if you look at all of these mutations in the poor, they're exceptionally conservative, right? I mean, the LVNV mutation, you're talking about a few methyl or methylene groups difference from what it is in wild type, right? So I signed on a little bit late, and sorry if this is a dumb question, but one, do you know that TTX is actually not binding as well? Do you have a dissociation constant, not a flux for these mutants? And the second is, why do you think those exceptionally conservative mutations are sort of enough to generate so much resistance?
So first off, you're correct on a few things. The a couple of these mutations just change a aspartic acid for a glutamine or a glutamate. We're talking just very, very small, minute changes, which suggests that those more conservative mutations in structure might be more for fine tuning, while others that change the hydropathy of the mutation might have an effect. So we'll get to that. But to your question of, do I have a direct binding constant? No, all I have is an apparent binding constant. I do have an idea for some experiments that we could use that is based on plasmon resonance. There's a new technique out called SPRM, surface plasmon resonance spectroscopy, where you basically take a cell, put it at the bottom of a dish, you angle some light at it, and it's got this gold coating, and the light excites the gold, and which is physically coupled to the channels or the proteins that are in contact with it. And as you wash on TTX, it's going to slightly change the weight of that, which is going to slightly change the signal that you receive. And that could give us a direct measurement of that direct binding affinity. Now, we could do that. Norm does have a turf machine, which is the basis for this, but there should be another graduate student that looks at that. Looking now at your question about why these tiny little mutations could cause a huge difference in the binding affinity of this toxin, I really like to think about this not just as the structure, but the properties of those structures. Here, what we're looking at right in the middle column B, that's the human voltage-gated sodium channel that was published in 2018. And I've mapped on here the hydropathy, or the fattiness of each amino acid. And we can see that band right there in the middle, that hydrophobic band of yellow. That's where it's going to sit in the membrane. Everything in blue is going to be more hydrophilic. If you look in A, that's a mutant that I have made. I've just superimposed all the differences that are found in snakes that are not found in humans. Jumping out of order then, if we subtract B from A, we look at C and we see all the points where the snake is more hydrophobic than the human. And we can see there are a few pockets of increased hydrophobicity there. And the brightest spot right under the C marker is that bright spot there. That's a part of the protein called the domain 1 voltage sensor. It's got this pocket of hydrophobicity. But due to the way that the structure of the channel is built, the domain 4 mutation, where we see LVNV, is directly deep to that. So these mutations, while in our experiments, which were based on rat channels, while we might not be able to suss out exactly how these work out, or exactly how a single point mutation leads to the greatest effect, I think if we were to instead work directly in snake channels, which has this hydrophobic background, we might be able to get insights that we didn't have when these papers were, to answer that question, were coming out about 10 years ago. So that would be the next step, would be working directly in the channel. There are some people who have done that, but they haven't worked on the same phenotypes that I've looked at here. So if that kind of answers that question. Cool. Nothing from the chat?
Doesn't look like. It's mostly congratulations, and I've gotten secret messages that we have to really grind you next.
Oh gosh.
Yeah.
So Robert, can I ask a question or sort of make a comment?
Oh, sure.
So there's actually quite a large theoretical literature on evolution on adaptive landscapes, and one of the sort of big issues is epistasis.
Mm-hmm.
And sort of the fact that epistasis dooms organisms to mediocrity. Stuart Kaufman coined that term quite a while ago.
Yeah.
So, I mean, this is incredibly elegant, really, really important work, but the reality is it's so much more complicated. And that is that you've got, other loci, and you've even got other sodium channel loci that could be doing compensatory or inhibitory things to this whole process. So have you looked at all at, gene expression, how these mutations change patterns of gene expression using, RNA-seq or things like that? Any students working on those sorts of issues?
Wow, thank you so much for the question. And what a wonderful shout out to Stuart Kaufman. In my first introduction chapter, I took a quote from the end of his very long 1,000-page book and threw that as the intro to my work. So I'm a big fan of him. You're right. This is so complicated. And the reason I frame this research the way it is because if you look at just this one protein, it supports a lot of the work that we've been looking at. But there are so many moving parts to the system. I mean, there were a lot of moving parts just in one protein. Understanding how it works when you incorporate other sodium channel mutations, their partner proteins, like the ones I mentioned for the excitation contraction coupling. There's so much involved there. And yes, our group as a lab does have students who've been looking in protein expression. So for instance, Jessica Remke, during her research, she did qPCR, she did RNA-seq, fantastic research that pointed out tons of potential pathways, at least in one species, that could be altered. And it is quite complicated. That work has yet to be replicated in these snakes where we already have a good understanding of where resistance comes from and its effects on the trade-offs. This work here really, it stands on the principle that these proteins, the voltage-gated sodium channels, have such a major effect on fitness that we can kind of briefly, compartmentalize our mind, put that under the rug for just a second to focus on this and its potential effects on evolutionary trajectories. But I think one of the best things that we could do, to your point, would be to start really assessing the whole genomes of all of these organisms and start mapping these things out. And there are students in our lab as well, Joshua Hallis, who will be working on these kind of phylogenetic changes that might point us in directions as well. So I do like the idea that epistasis dooms things. And again, just what's on the screen right now is pointing at, even within a protein, you can't take anything for granted. Even if the pore of this protein is conserved from flies to humans, the background of that channel may influence the molecular epistasis that we're seeing just within this protein. So yes, lots of complicated things that I would love to see come to fruition in the future.
All right, thanks.
No
Great
problem.
job.
Thank you for tuning in.
Bobby, I have a question following up on that, actually. Hang on.
Okay.
I guess I have two questions, but the first of which is, I understand a while ago there was that paper that came out about looking at the parallel evolution of TTX resistance and seeing that it didn't arise in the what was it? It was only in the peripheral channels and not in the ones that were not exposed to TTX. I'm not sure if I'm really articulating that correctly, but yeah, because of the blood-brain barrier, I guess, and the inability of TTX to cross that. Has there been any follow-up work on that, and is that reflected in the populations that you've been looking at?
So that's actually a great point. You're bringing up something really interesting, and it's actually the subject of one of my appendices that Norm was talking about in my dissertation. The sodium channels in our snakes that have developed resistance are 2 found in peripheral nerves of the snakes and 1 found in the voltage-gated sodium channel in the skeletal muscle, like we've seen. Now, those are the ones that are going to be directly affected by TTX as it enters the bloodstream during a predation event. And so it kind of makes sense that we would see some changes to those mutations, and we do. And we don't see any mutations beyond that. Principally, the sodium channels that evolved on the other side of the blood-brain barrier where TTX can't access it, they're going to kind of evolve independently of any selection pressures as long as the TTX concentrations don't get high enough to cross and drive through that membrane potential. Or excuse me, that's just a word I throw out now, across that membrane. And as it happens, a couple of them randomly have a little bit of TTX resistance, but it's not, not evolutionarily significant for here. But one thing that I would like to point us to your question, is that we have sodium channels in our heart, and across phyla, those cardiac sodium channels are natively resistant to tetrodotoxin. They have a point mutation in the first domain of their channels, a cysteine mutation, that makes them resistant to the toxin. But now our snakes, these skeletal muscle voltage-gated sodium channels have mutations that are so extreme that no newt could produce enough toxin to block them. That now my thought is the cardiac sodium channels are going to have to keep up with the skeletal muscle sodium channels, and the peripheral nerve sodium channels are also going to have to try and keep up with this extreme resistance, because eventually these newts are going to start trying to amp up their toxic load as best they can, and this pressure might become real. So to this idea that, you know, of the 10 paralogs of sodium channels that we see in the human, or excuse me, in the snake, when one of them responds to an evolutionary challenge, it's possible that it may drag the rest of them along with it. So I hope that kind of answers your question. But to be honest, I don't know anything further. I know that there is a former student of UNR who's at Virginia Tech who has been looking at those in other species like herons in the Florida coast. So.
Cool. Yeah, thank you. Yeah, that does answer my question. And I remembered that as being an interesting example of kind of the predictability of this system, which is really, yeah, it's incredibly fascinating. But yeah, my second question was earlier on when you were showing the diagram of the human voltage-gated sodium channel, and you were showing some of the mutations that were maladaptive.
Mm-hmm.
I was just curious, what were those maladaptive with respect to? Like what trait?
Okay, yeah. So let's jump back here for a little bit. So we have the human, or excuse me, I keep wanting to talk about humans. I don't know where that word's coming from. So this is Thanophysa atratus. This is one of our species. And these are the 3-point mutations that are mutated in its species. I believe that A1281P results in a little bit of TTX resistance. It's not shown here, but that's going to create some ring strain. It's going to add a little bend to that part of the ribbon that is going to kind of change the access of the toxin to the pore. And that giant bend may also affect how this protein conducts forces throughout itself during the conformational changes that it needs to activate and inactivate. D1568N, we only observe that mutation in extremely resistant animals that are also biophysically compromised. So my thought is that mutation, which seems like it shouldn't, if you know the structures of those amino acids, it shouldn't make too much of a change, but it is a charge difference there. That could change the way the pore works. Going back to that idea of hydropathy, the hydrophobic or polar substances in the protein, that might change the way the protein slides against itself or how it conducts those forces as well. But D1277E, this is a D to E mutation. That's only, it's so similar, right? There's only one methyl group difference between those two. So I'm not, I really think that is just a fine tuning mutation trying to, reform the structure of that protein. If we look at LVNV, if we look here, so this is LVNV. These are our 4-point mutations in this snake. D1568, sorry, I lost. You're asking about the human ones?
Yeah, I was just curious. I mean, in general, like, maladaptive would suggest like.
Are you talking about this part here?
Yes.
Oh, okay, I'm sorry. I was confused. So we know most of these mutations result in a stillborn. The vast majority of these mutations are so devastating on their effects of human. The entirety of the sodium channels in our genome is only 0.041% of the entire genome. If you make a single mutation to that very, very small proportion of our genome, it's likely that you're going to have a stillborn animal. Some of these mutations are less severe. Some of them are going to be less severe and result in something called periodic paralysis, where under certain circumstances your sodium channels are just a little sluggish to get moving. When we compare where we find these sodium channels mutations in the snake relative to the human, you can kind of see that the humans, at least, we really avoid the poor, where we're seeing poor mutations there. You see a lot of yellow where you wouldn't see any mutations that crop up in humans, which suggests that they're so costly they don't even get off the ground, right? We don't even observe them in humans. They're that well preserved in humans. But some of them are a little bit closer to others. So there are some really strong evidences just looking at this to say that any mutation is costly.
Wow. Thank you.
Yeah, no problem.
And yeah, again, fantastic talk. Just unbelievable.
Okay, I appreciate that. Thank you so much. Oh, one from the chat. Okay.
This will be the last question, Robert. We
Okay.
need to move on to the
Yeah.
actual defense.
Okay. So this last question here is about how these mutations play out in other species. Talk about repeatability. We see these mutations crop up in the very animals that that produce this toxin, right? They have to survive. They have to survive their own chemical defense. So pufferfish and newts and blue ring octopus, they have their own mutations and their own ways of dealing with this toxin that are surprisingly repeatable. We see these mutations like the D, the E, the N, the L, the V. They're everywhere wherever these sodium channels are facing this evolutionary challenge, this physiological challenge. So my personal thought is that any animal that has these chemical defenses can afford to be slow and weak, right? Things are gonna avoid you if you are so toxic. So you can afford to be slower. You can afford to take these evolutionary hits and mess up your sodium channels a little bit because you have that protective factor. And there was a second part to that question. Oh, right. So is there a benefit to the snake for accumulating TTX? There is some evidence to suggest that after a snake has been fed a newt, 30 days later, their livers are still loaded up with TTX. I think it's on the order of micrograms of TTX that's still in there, which is still quite enough to paralyze a higher predator, like a heron or something that would want to eat those snakes. But here's the thing. We also know that our most resistant animals may have this difference in coloration, that they may be protected enough to start incorporating colors to their physical being that make them a little bit more visible. It's the idea that you're saying, hey, don't eat me, I'm toxic, right? It's kind of stay away from that. So who was that question from? Oh, Brian, thank you so much. All right, so I think that was our last question. Anyone who has any questions, you can always reach me by social media and I will answer these as much as possible. And I wanted to thank everyone around the world for joining in. Thank you so much.
You did a great job, Bobby.
Thank you. I see all you clapping.
Thank you all.
Okay, Norm.